Well, thank you, Jess, for a kind and flattering introduction, I think. It's a wonderful honor to get the Urey Medal from the European Association of Geochemistry. Having one's name associated with that of the great Harold Urey is as much of an honor as anyone could wish. It's particularly flattering for somebody like me who has not used isotopes as a main tool to probe processes at the Earth's surface. Following many others, what I have attempted over the years is to understand geochemical processes by looking at the underlying fundamental chemical and biological mechanisms. As an example, I'll tell you the story of cadmium in the ocean as it has unfolded in my group over the years as a result of the talent and hard work of many students and postdocs who, of course, deserve to have their name associated with the Urey Medal. As you know, when we stand here, usually the work of others is what's being honored, and so I want to underline that many times. And as I talk, I'll mention the name of a few people. All right, so I don't know how much time I have. Well, it's roughly at the right time. Okay, so I'm going to tell you about the geobiology of cadmium. I have a pointer somewhere. Maybe not. Maybe yes. That's very good. And this talk, of course, is attempting to make a junction between molecular processes and global processes, which is a quest that's been going on in my group for several years. And this story starts somewhere in the mid-70s with Ed Boyle's thesis at MIT. Ed Boyle did some of the first measurements of trace metals in the ocean and discovered this extraordinary behavior of cadmium in the ocean, which has essentially zero concentration at the surface, going up at depth, and parallel exactly that of phosphorus. Phosphate has exactly the same kind of profiles. And this behavior, of course, is what we call nutrient-like profiles, and it implies something. I see Ed Boyle over there, so this is work replotted compared to the first papers. And this correlation between cadmium and phosphate has really stood the test of time and has been the basis of work having to do with using cadmium in foraminiferous to look at phosphorus in the ancient ocean. For me, it was interesting because if you look at the metals that are the elements that are thought to be useful for the biota, you'll find many trace elements of interest to me, but you don't find cadmium typically, which has been considered a very toxic element over the years. And so this behavior of cadmium in the ocean brings up the possibility that perhaps it's actually a metal that's actually used by the biota. And that, of course, would be an interesting result. The first work in my lab that addressed that issue was done by Neil Price, who is now at McGill University. And the idea is very simply to grow an organism, this famous TW that we talked about, one of those organisms that are thought to take up the nutrients at the surface of the ocean and bring it down in depth. And here is a plot of the log of the number of cells as a function of time, so it's simple growth curves, and you see pretty good straight lines, pretty good exponential growth. And in green is a normal growth medium with sufficient available free zinc, 15 picomolar. If you lower that zinc to 3 picomolar, you find much less growth, much slower growth. And if you add back cadmium in the medium, you regain the growth rate. It's an interesting phenomenon. It implies that cadmium can replace zinc in some biological function. And it turns out that this phenomenon is particularly visible when you have low carbon dioxide in the medium. So right there you have an idea that if cadmium is going to be useful, it has to do with some function that zinc often can play in the biology, and also it has to do with something having to do with carbon dioxide. So the idea that came out of that was to look for an enzyme that might actually be a zinc enzyme and works with carbon dioxide. And the obvious idea is going to be carbonic anhydrase. The reaction that you have all studied in your life in bicarbonate and CO2, which we often take to be very rapid in natural waters, is actually quite slow, something like half a minute or so to equilibrate. Well, that's much too slow for cells. the time it takes for things to diffuse in cells, a lot of microseconds or whatever, it won't happen on the order of half a minute. And so biology has invented a wonderful enzyme called carbonic anhydrase, known as the zinc enzyme, and which instead of something on the order of half a minute, we're now down to the microsecond scale. And so this is a very common enzyme present in all organisms we know about, pretty much. And so we set out to find this enzyme in our model organism, and those are all slides of protein gels, non-denaturing protein gels. On the left, you have an inlay assay where we actually look at the color change to measure the extent of carbonic anhydrase activity. And on the right, you have a measure of the zinc 65 in the same proteins. We have grown the organism with the radioactive zinc, and we can then find the radioactive zinc in the protein gels. And what you find is, it seems essentially in the same proteins, much of the zinc where there is activity, and very little zinc in aple cells. So that means that this enzyme likely contains a very large fraction of the cellular zinc, something a bit unexpected. And you'll find that the lanes which have a lot of activity and a lot of zinc are those for the organisms grown at low carbon dioxide, which increases apparently this expression of the carbonic anhydrase enzyme. So that's very good. And so we continued that work, and then we ended up finding an enzyme. The sequence of the amino acid is right here, just to remind myself to tell you that it's found a new class of carbonic anhydrases. We didn't find that particular kind before. And we were able to do a couple of things with it. One is to get enough to actually do some X-ray spectroscopy on it and to characterize the zinc binding center. And what you find here is the nitrogen binding of the zinc with the water. And it's a characteristic of all the alpha carbonic anhydrases, which are very common in animals. We were able also to actually overexpress this enzyme in E. coli, not in an active form, but in a form which allowed us to have an antibody. And the antibody is convenient to do some experiments, so I'll tell you about those. And here is a typical experiment. So on top you have, and it's going to be all those bars are measures of whole cell carbonic anhydrase activity. The second panel is a measure of the concentration of this enzyme, which we measure using the antibody. And here you have a set of conditions in which we vary the metals and we vary the carbon dioxide. And what you have in the first three here is full zinc in the culture and varying CO2 from low to higher CO2. And so, so you have high activity at low CO2, lower activity at high CO2. The second three bars here shows the low zinc cultures, same thing as a function of CO2. So again, an effect of CO2, but much less activity because we have much less zinc and much less enzyme. And indeed, we have less enzyme as we can measure through this antibody. We do the same experiments adding cobalt to the culture. We regain our activity, and we regain the concentration of that enzyme. So this organism can replace zinc by cobalt in vivo to regain the activity of the enzyme, replaces the zinc by cobalt in the enzyme itself. When we tried what we thought was going to work, which of course we put cadmium instead of zinc, and sure enough, we got activity back as we thought we would, but we didn't get any of the enzyme back. And so it was the wrong enzyme, which is the wrong thing. And what we did find is some places on the gels with a lot of cadmium-109 activity when we labeled the cells, and indeed it responded to CO2, but it was not this particular enzyme. So that was a lot of work at that point because we had to essentially isolate the cadmium enzyme from cultures. And for those of you who try to do cultures under fairly clean conditions, the idea of having very low zinc in cultures and very large amount of cultures is, of course, a bit of a formidable task. And so some postdocs spent a lot of time on that, and Max Saito was here, was much involved in this work. And we ended up being able to purify this enzyme. You can see some purification here on these columns, and you can see, in fact, the CA assay, which is working with nothing else, the Kumasi showing that it's only one protein, and the phosphor image showing that actually there's cadmium in it. And that allowed us to actually get the full sequence of the enzyme, which turned out to be really annoying because essentially the blue, the red, and the green are exact repeats of each other. This is actually a trimer, and I'm anticipating on what's going to come, but actually any one of those monomers is actually a fully functional enzyme. The fact it was a trimer in our organism makes it very difficult to actually sequence properly. Again, it was a new enzyme, and we had very little of it, of course, because it's hard to isolate. And so we did some X-ray spectroscopy at Stanford again, and it's quite noisy because you have very little material. But it was enough in terms of the Xanes data to be able to show that it was very much like some of the sulfur complexes of cadmium, not some of the others. And on that basis, we were able to conclude that the coordination of the cadmium in the enzyme was not at all like that of the alpha CA that we had found before, which we call a delta CA. It was a new family. In this case, this new family of enzyme really looked like the beta CAs that's found in many plants, in which you have 2 sulfur and 1 nitrogen through cysteine and histidine doing the binding in addition to the water, which of course is necessary for the reaction to take place. All right, and so at that point we have some results, we have an enzyme, we have cadmium, and the question becomes why bother? Why should organisms bother with using cadmium in the ocean? There's not very much cadmium there, there's much more zinc. Why is it so important to have carbonic anhydrase? And so I'm going to make a detour here to the question of the inorganic carbon acquisition in seawater. Those organisms that do photosynthesis must get CO2 to do photosynthesis, and the issue is how do they do it, and why might that be a problem? And so here is a plot that I love because I don't even know what it means on the y-axis here, but it's something having to do with what organisms were there when, as a function of time over 355 million years. The point of this is to show that the greens disappeared from the ocean a long time ago, pretty much. There's very little of it in the ocean now. But there are things like diatoms, which have taken up over the photosynthetic activity of large, the eukaryotes in the ocean, really came up starting, I don't know, somewhere here, we're not quite sure, and really taking over the last 50 million years. They're very new organisms. They're organisms that have arrived to do photosynthesis on the planet very late. And so if we go back to the kind of work that Bob Berner, who might be here, actually did, well, we know something about the change in carbon dioxide on the planet as a function of time. And we know that it used to be high. This is a huge range of errors, of course, but 400 million years ago it was very high. And that's when we had, of course, a lot of green organisms. And it's been much, much lower since. And again, maybe up and down, but by and large, much, much lower. So it's my sense that, in fact, those organisms that took over late took over in a world with very low carbon dioxide. And the problem then becomes that of the fundamental biochemistry of photosynthesis, of carbon fixation. Rubisco is the enzyme really doing the job, responsible for fixing the carbon in photosynthesis for the dark reaction. This old and huge enzyme, maybe the most abundant in the world, everything that's green pretty much has it, is quite inefficient. And so this is a cartoon I made a long time ago. Rubisco is a magnesium enzyme. It's a metal enzyme with magnesium. The bisphosphate is here, the CO2 is here, and that's in the process of doing the fixation. Now, the affinity of Rubisco for CO2 is very poor, which makes sense. If the enzyme existed hundreds of million years ago, there's no need to have a great affinity. Then it's a very slow enzyme. 3 per second is not exactly remarkable for catalytic activity. And finally, it's become poisoned by oxygen, which has been evolved by blue-green on our planet. And so here we have real trouble in fixing CO2. And so a number of organisms have evolved means around that. And diatoms, we think, have evolved a particular system to acquire inorganic carbon. We call it carbon concentrating mechanism, CCM. And this is the work of John Weinfelder, who probably is here as well. He's now at Rutgers University. And discovered pretty much the system that exists in diatoms, which looks very much like that of C4 plants. You can follow these arrows here. You go from bicarbonate in the water, being pumped into the cell. This is a diatom with a frustule and a cell membrane, pumped into the cell. There, bicarbonate reacts with an enzyme called PEP-C to become a C4 organic compound, goes into the chloroplast, it gets decarboxylated by another enzyme, the CO2 is fed to Rubisco, and of course, that's where the actual photosynthesis, the actual carbon fixation occurs. I won't show you the original data of John Weinfeld. I'll show you some newer data showing the importance of those two enzymes in the actual photosynthesis of diatoms. Those two enzymes here. And this work of Patrick McGinn, who is now back in Canada. And here is the effect of inhibitors of PepC and PepC-K on two model diatoms and two model green algae. You see that the greens are completely indifferent to the presence of those inhibitors, while the two diatoms are completely crippled by those inhibitors. It's simply unable to photosynthesize when we block those particular enzymes, which are useless pretty much, or secondary importance in the greens. I hope that convinces you that diatoms are not simply green algae in disguise with some frustules around them. Let me go back to this cartoon here and talk a bit about the carbonic anhydrases. Why are they there? Well, they do the rest of the business. The carbon comes up and be accumulated here, but then it's going to leak back out. CO2 goes right through biological membranes, and so we're going to have leakage of the CO2 going outside. So this carbonic anhydrase here in the cytoplasm actually equilibrates the CO2 with the bicarb. Since the bicarb is very low because of this PepC activity, the CO2 is kept extremely low, right? The pH is about 7 inside the cell, 7.4. And so we have a very low CO2 here. So this leakage of CO2 is recycled back into the system. There's an external carbonic anhydrase, and this one is interesting because bicarbonate can have no diffusion gradient. There's much too much bicarbonate in seawater to have any diffusion problem, but CO2 has a diffusion gradient, and so this carbonic anhydrase allows this forces an equilibrium between the bicarbonate at the surface of the cell with the CO2, ensuring the maximum value on the outside of the membrane. Hence, there's a gradient inward of CO2 instead of outward. So those two carbonic anhydrases really play the rest of the game in this CCM system of diatoms. And what we find, for example, is that when we go from high to low CO2, we can see, this is a reverse transcriptase experiment, in which we look at the amount of those enzymes being expressed in the cell. You see that immediately as you lower the CO2, you have a jump in expression by manyfold. And it is the enzyme that we find to really change when we change the CO2 in the medium. I'll do a small parenthesis here because many of you are interested, I know, in things like formation of diatom frustules or the calcite of coccolithophores, and so I'll make a small story here. This external carbonic anhydrase is interesting because carbonic anhydrase requires a buffer to work. The protons in seawater are not enough to actually allow it to work, and so you always need to add a buffer to get activity. And this is a typical scheme of carbonic anhydrase, where you have on the one side, you see that the bicarb CO2 reaction on one side and shows that the proton goes around for that. And on the other side, essentially, is the recharging of the enzyme by acquiring a proton from other residues on the enzyme and making it happen. Well, this exchange of the proton here requires absolutely a buffer. And so the problem of buffers in seawater, that they are much too high a pH. Bicarb is not actually useful for this reaction, and so all you have left is pretty much the borate system, which pK about 9 or so. And so we wondered whether perhaps a frustule might actually play the role of the buffer, because silica has many nice properties. And we did some experiments a while ago, and maybe it's true. It's possible. You see here, this is the carbonic anhydrase activity. It's a very simple assay here. In the phosphate buffer, then no buffer, and adding various silicic acid concentration, and then adding some preparation of frustules, and we regain the activity. So I think it's conceivable, actually, that the frustule is served as one thing among many, I would think, as a acid-base buffer to allow some enzyme to work on the outside of the cell. So far, what do we have? We found an enzyme which contains cadmium, and we may have a reason why it might be important because of the inorganic carbon acquisition. And at that point, we got lucky. Yan Zhou, who is a postdoc in my lab, had been doing other projects, and secretly she was working actually on this cadmium carbonic anhydrase. Indeed, she didn't tell me about it, but one day she said, you know, I've got it overexpressed in an active form. And so she had lots of cadmium carbonic anhydrase overexpressed in E. coli. And so as soon as she had that within weeks, that collaborated with Yongxi at Princeton before I went back to China, and we had a whole bunch of actual crystals of the enzyme. So we could do all kinds of very fancy characterization down to 1.4 angstrom. And so part of the story now has to do with this new revelation of what the enzyme really looks like and what it can do. By the way, this is a picture of the enzyme. I'm very fond of those ribbon pictures of molecules. I think they're quite pretty. And so how does the CDCA work? So here is pretty much the metal binding center of this enzyme. CDCA is in green, and superimposed in blue is this beta carbonic anhydrase, which has been characterized from Pisum sativum, the English peas, which is the only one that has a complete, really nice X-ray crystallization characterization. And so here, the superposition of the active site, and you see not only the two cysteines and the histidine, but you see also an arginine aspartate pair with a kind of adjoint bonding, which is essential to the functioning of both the beta-CA and the CDCA. And the superposition is quite remarkable. If you mutate any of those residues here, here's the wild type activity, here's the mutated activity, there's nothing left. It doesn't work if you get rid of those amino acids, of course. And so here is the actual binding distances for the metal in the enzyme. And what you see, of course, is the binding of cysteine and histidine, but you will see also here is the water binding. And it's an extensive network of adjoint bonding of water molecule. And that, of course, is the key to making carbonic anhydrase work. You have to change the acid-base chemistry of the metal to allow this proton exchange. And so the whole business of carbonic anhydrase is to have a coordination that manages to change this nice water of coordination, how much it's hydroxyl, how much water, how facile is the exchange, the actual pKa of that water. Now this mimicry between our CDCA and this beta-CA from English peas goes further than just this metal binding center. It actually goes fairly far out. This is superposition of the two. And particularly here, what you can see is that we have this metal binding center, and here we have other residues which are also well conserved, and they're involved in substrate binding. And been shown in the English peas. In our case, we were lucky enough to get a crystal in which with acetate instead of CO2, it crystallized very nicely and characterize actual binding of the substrate. And so those are involved in substrate binding. And you see the similarity. Phenylalanine replaces another one, the tyrosine replaced by leucine and threonine, and it really gives the same sort of conformation to the enzyme. What's extraordinary is that we have a monomer in our case. In the case of the beta-CA, it's a functional dimer. The enzyme does not work unless you have a full dimer. And that's because some parts here are on this other part of the enzyme, the other dimer. And so I think it's a perfect example of converging evolution. Completely different enzymes, evolving completely differently, ended up with a very same solution. The metal center becomes the same because that's the only way it's going to work. I think it's quite remarkable. Well, lo and behold, what happened? Well, E. coli was very happy to put zinc inside the enzyme. We didn't force it. If we didn't put cadmium in it, put zinc in it. We have a very nice zinc enzyme. We have the binding of zinc. And we can demonstrate an easy exchange of zinc for cadmium. That's a bit of a complex slide. Here we're measuring the exchange of zinc, which was in the enzyme for cadmium, supplying cadmium in two different forms, an NTA complex or a phytochelatin 2 complex. Phytochelatin is an interesting peptide, which is a gamma-glucis, in this case twice with the terminal glycine, and which binds cadmium very nicely inside cells, as we know. And so we try to put cadmium in that form and see if it gets into the cell, and sure enough, it gets into the cell very nicely. This is a control experiment with a beta carbonic anhydrase, which we overexpressed, got from and overexpressed from Phytoplankton trachonutum, another diatom. It's a real beta-CA. You see, it doesn't do the job. It doesn't exchange nearly as well the cadmium for the zinc. And in the case of this one, we cannot do the reverse reaction because we can't get the cadmium form, but here we could do it with zinc getting into the cadmium form of our enzyme, and it exchanged very nicely in the case of the phytochelatin. In the case of NTA, it doesn't exchange at all. And that's because the relative affinities of zinc and cadmium are not right if you use NTA. NTA is much too happy having zinc and not happy enough having cadmium in it, while phytochelatin can do both. And so, in fact, we wonder whether phytochelatin might actually be used inside the cell as a buffer for cadmium, because we know cadmium binds inside the cell to this phytochelatin. And why does it exchange? Well, so we were lucky enough to get another crystal without the metal in it. And so this crystal is deformed. As you see, here's the binding center with the metal. The metal is gone here. I put it here just for distance. And this thing completely opens up. This metal binding center opens up. There's a 9 amino acid sequence here between the cysteine and the histidine, which completely changes configuration when there is no metal in it. And so we have this sort of hinge coming in and out of the system. And we wondered about that. And so we said, is that true? Is that really important? And so here is the sequence of that hinge from the histidine to the cysteine. You see you have glycines just flanking both things here. And glycine is known as a very flexible amino acid which allows things to move. And so the beauty of this sort of experiment, you can actually mutate this glycine, one of them, the other one. No, one of them, the other one. And if you mutate them, of course, you can't exchange the metals anymore. And so you can prove directly that this particular glycine hinge is essential for metal exchange. And so we have here an enzyme which has been really made for exchanging metals, which I find interesting. A true chemialistic enzyme. Mike Saito is very fond of the idea of a chemialistic enzyme. Nobody knows what the word means. It means exactly that. It can exchange the metals. All right, so does this enzyme actually work? So we did a little bit of kinetics. Here is the method for the kinetics, and see, with geochemists, we love to use isotopes, of course, a little bit. And so we have triple labeled CO2, and we're adding the enzyme. And what you see is the mass 49 becoming, of course, I can't read it over there, but it should be mass 45 over there, mass 47, which appears for a while. Essentially, the oxygen is changing without of water, and you can follow that very nicely. And so here is the Kcat over Km of the enzyme, which you can think of as a second order rate, okay, as a function of pH. And what you see are numbers for the zinc enzyme, which are nearly 10 to the 9 molar minus 1, second minus 1, and for the cadmium enzyme, a few times 10 to the 8. Those are very, very fast reaction rates. We're very close to diffusion limit. It's about as fast as enzymes can work. And so we have an extremely efficient enzyme. Even though it's more efficient in the zinc form, we can show it actually quite fast enough in the cadmium form as well. And so that's a lot of fun going into the molecular part of this game. And now we are geochemists, we are oceanographers, and we like to go back to the field. And so does it matter if we find this enzyme in the field? We find it anywhere. And so we looked at it, looked for it in a number of cultures, so the number of cultured organisms, the Theoceanica, other things, Trochonitum, and a bunch of field samples. And everywhere we looked, we found that enzyme, often several forms, and very well conserved. This is a tree, biologists love to show that, where the distance here shows how many amino acids have been changed, but in fact, it's extremely well conserved. And so this enzyme is found absolutely everywhere and very well conserved. Is it actually used in the environment? Well, so this is New Jersey coastal water, the best there is. No? All right. And so very simple experiments. You incubate the water and pH changes, and there you can make it vary if you want. You can measure the carbonic anhydrase activity. Of course, it increases at higher pH, at lower CO2. You can look at the expression of the cadmium carbonic anhydrase, and it goes up at higher pH. And you can measure the enzyme itself, again, using an antibody, and it goes up as well. And so it's pretty clear this enzyme is actually there, it's actually used, it's actually quite active. What we have not been able to do is to quantify in any way yet the amount of cadmium actually used in seawater, in the ocean, in this enzyme. That would be, of course, what we'd like to see, that in fact, that is important in the geochemistry of cadmium. So we don't have that yet. We're working on methods to do that. But what we have is an older experiment, which I think is the kind of thing we often do in science, where we can't really prove what we want to prove, but we can make predictions, and if they come true, then we feel better about things, right? So here we are going to look at the effect of zinc and CO2, and I'll show you the CO2 data, on the amount of cadmium taken up by cells, right? The prediction, of course, is that low CO2, they should take more of the enzyme, more cadmium per cell, et cetera. And so this is a work done of Morel, and for those of you who are not West Coast types, there is an interesting sort of behavior of seawater there, which comes underneath, goes to the surface at the coast, and goes back out. And this high CO2, high nutrient water loses its CO2 and nutrients as it goes further offshore. And so you have a natural gradient of CO2. And so here is a plot. It's worked with Jake Cullen, at the time was a student at Rutgers University with Rob Sherrill. And what you have here is a concentration of cadmium normalized per phosphorus inside the cells, which are full of diatom blooms, as a function of the CO2 in the water. And as you have, of course, this high value at low CO2 and lower value at high CO2, as we had expected. Now, one can play some games with that, and you might decide whether they're useful or not. Here's a global sort of correlation between cadmium and phosphate in the world, and I'm going to be very rough, but I'll put a straight line through that. I'll simplify the problem and tell you that the slope is on the order of 0.3 millimole per mole cadmium per P in seawater. It's sort of a Redfield ratio, if you want, of cadmium per phosphorus. What happens to what's happening in this? What's about the cells? Well, if we take something like atmospheric CO2, we look at where we are in our CD to P ratio here, we get somewhere here, which is on pretty close to 0.3. So I'd like to say that this cadmium to phosphate ratio is in the cell. It's very much like that in the ocean, and it has to do with the activity of this enzyme. You don't have to believe me. It's a nice story right now. Okay, so that's as good as it's going to get for a while. Here are the people doing the work, and I've mentioned a few people here. Pat McGinn, who did some of the CCM C4 work. I mentioned Ewan Park, who did some of the coastal work in New Jersey, and of course Ian Zhu, who did all the fancy work of overexpressing the enzyme and getting the crystals. And I thank you.